Right this very second, there are millions of cells in your body dividing, each one creating a perfect copy of itself. It's the process that literally built you from a single cell, and it's the same one that's healing that paper cut on your finger. But how on earth does a cell flawlessly copy and distribute its entire massive operating manual, its DNA, without messing it all up? Well, let's dive into the incredibly elegant and precise stance of the cell cycle. So, here's our roadmap for this explainer. We're going to start with the big fundamental idea of where cells even come from. Then we'll zoom way in to see how DNA gets packaged up for its big move. After that, we'll watch the step-by-step performance of division itself, uncover the tiny molecular clock that runs the whole show, and finally, we'll look at what happens when this beautiful system goes wrong. More than a century and a half ago, a physician named Rudolf Virchow nailed a huge biological truth with this one simple Latin phrase. It means every cell comes from another cell. All life is just this continuous, unbroken chain of cells dividing. It's the foundation for literally everything we're about to talk about. Okay, so why is this process of one cell becoming two so unbelievably important? Well, its jobs are about as fundamental as it gets for life itself. For a single-celled organism, say an amoeba, cell division is reproduction. That's the whole shebang. It makes a brand new individual. For complex creatures like us, it's how we go from a single fertilized egg to, well, us. And it never stops. Every single day, your body is using cell division for renewal and repair, like your bone marrow, which is just churning out millions of new blood cells to replace the old ones. Now for the central puzzle. Before a cell can split, it has to make a perfect copy of its genetic blueprint, the DNA, and then, this is key, it has to hand out those copies perfectly, one to each new cell. This is just a monumental feat of biological engineering. To give you some perspective here, get this. If you stretched out the DNA from just one of your cells, it would be about 2 meters long. That's taller than most people. And all of that has to be crammed into a nucleus that's about 250,000 times smaller. So how is that even possible? The answer, the brilliant solution, is the chromosome. Just think of it as the ultimate data compression and storage device. The cell takes that super long DNA molecule and wraps it tightly around these proteins, coiling and folding it with mind-boggling precision until it's this compact, manageable little package. It doesn't just solve the storage problem, it makes the DNA easy to move around without getting tangled. So let's follow the process. A cell usually has its DNA as its long, thin, kind of messy fiber called chromatin. But to get ready for division, it first duplicates that entire strand of DNA. Then, the original and the new copy condense down super tight. And what you get is that classic X shape we all recognize. That's a single duplicated chromosome. Each half of that X is an identical copy, called a sister chromatid, and they're joined right in the middle. They're perfect twins, ready and waiting to be pulled apart and sent to their new homes. And now we get to the main event, the actual physical process of division. And this isn't some chaotic explosion, it's a beautifully choreographed step-by-step dance. So let's break down this amazing performance. First thing you need to realize is that the dramatic part, the division itself, what we call the mitotic phase, is actually the shortest part of a cell's life. About 90% of the time, the cell is just hanging out in interphase. That's when it's growing, doing its normal cell job, and most importantly, copying all of its DNA to get ready for the big show. The show begins with prophase. Inside the nucleus, those long, stringy chromatin fibers start to condense into those thick, visible chromosomes we just talked about. At the same time, outside the nucleus, this amazing structure called the mitotic spindle starts to form. This is the machinery made of tiny tubes that's going to do all the heavy lifting. Next up, prometaphase. The wall around the nucleus, the nuclear envelope, just disintegrates. It breaks down. This clears the stage, so to speak, and allows those spindle fibers to reach in and literally grab onto the chromosomes. They latch onto a special protein handle on each sister chromatid. Now, this stage here, metaphase, is absolutely critical. You can think of it as the cell's big quality control checkpoint. The spindle fibers play this microscopic tug of war, pulling and pushing the chromosomes until every single one is lined up perfectly along the cell's equator, a line we call the metaphase plate. The cell will actually pause and wait until this alignment is absolutely perfect before moving on. And then, bam! Anaphase. The go signal is given, and an enzyme just slices through the proteins holding the sister chromatids together. They're instantly pulled apart, and now they're considered individual chromosomes. Each set gets reeled in toward opposite ends of the cell, like they're on a fishing line. This ensures each new cell gets one complete set of the genetic instructions. In the final act of mitosis, telophase, the cell basically just hits the rewind button. Two brand new nuclear envelopes form around the two separate groups of chromosomes. The chromosomes themselves start to relax and uncoil, going back to that stringy chromatin state. At this point, the division of the genetic material is officially complete. But hold on, there's one last step. It's called cytokinesis, and it's all about splitting the rest of the cell. In animal cells like ours, a ring of proteins acts like a tiny drawstring, pinching the cell membrane until it splits into two. But plant cells have that rigid cell wall, so they can't just pinch. Instead, they build a new wall right down the middle, called a cell plate, that grows outward until it completely separates the two new cells. Okay, so we've seen the mechanics, the what of cell division, but how does the cell know when to do all this? The timing isn't random. It's all run by this incredibly sophisticated molecular control system. It's like an internal clock making sure everything happens exactly on schedule. I love this analogy from the source material. Think of it like a washing machine. It runs on an automatic timer, right? But it's smart enough that it won't start the spin cycle until a sensor confirms that all the water has drained out. The cell cycle is the exact same. It has this built-in clock, but it pauses at those checkpoints we mentioned, waiting for an all-clear signal before it dares to proceed. One of the star players in this control system is a type of protein called a cyclin. And just like their name suggests, their concentration inside the cell rises and falls in a very predictable cycle. When their levels get high enough, they basically act as the go signal for the next phase. So the cyclin is the signal, but what's the engine? Well, that would be an enzyme called a cyclin-dependent kinase, or CDK for short. These CDKs are always floating around in the cell, but they're inactive, they're switched off. When a cyclin comes along and attaches to a CDK, it's like turning the key in the ignition. The activated pair then goes off and turns on all the other machinery needed for the next stage of the cycle. This control system is normally so precise, so robust. But what happens if it fails? What if a cell loses control of its own division and just won't stop? Well, the consequences can be absolutely catastrophic. This is the very basis of cancer. This slide shows two of the most critical rules that cancer cells break. See, normal cells have good manners. They show what's called density-dependent inhibition. Once they form a single layer and touch their neighbors, they stop dividing. They also need to be anchored to a surface. Cancer cells, they completely ignore both of those stop signals. They just keep dividing and dividing, piling up on top of each other, forming a tumor. And because they also lose that need to be anchored down, some cancer cells can actually break away from the original tumor. They can get into the bloodstream or the lymph system and travel to totally different parts of the body, starting new tumors there. This dangerous spread is called metastasis, and it's what makes a cancer truly malignant. When you think about it, from growing us out of a single egg to constantly renewing our bodies, the cell cycle really is the engine that drives life itself. Understanding its tiny details, the packaging, the dance, the controls, is absolutely critical. In fact, many cancer chemotherapy drugs work by jamming the gears of this engine in those rapidly dividing cancer cells. It's a powerful tool for sure. So the big question for the future is, as we get better and better at fixing the bugs in this fundamental code of life, what other diseases might we finally be able to conquer?